Okay.
Welcome everyone to this new edition of the LIBREhub seminar. As you may know, in our open hardware project, we are teaching how to build instrumentation with low cost for microscopy, and computational methods are important because nowadays more and more techniques use complementary complexity in the computation in order to gain what's more simple in hardware. So today, let's welcome Dr. Shwetadwip Chowdhury. He's an assistant professor and fellow at the Jack Kilby and Texas Instruments Endowed Faculty Fellowship in the Department of Electrical Engineering and Computer Engineering at the University of Texas at Austin. Dr. Chowdhury earned his PhD in biomedical engineering at Duke University and was an NIH Ruth Kirchenstein postdoctoral fellow at UC Berkeley. He's a recipient of the NSF Career Award, and he will talk about his research on computational influence scattering models that can reconstruct quantitative 3D images from deep complex samples. So Shwetadwip, thanks so much for joining us, and the floor is yours.
Okay, thank you. Thank you all for having me here. It's really exciting to give this talk. I'm a big fan of your YouTube channel. Actually, my students are starting to look at your YouTube channel. Which I think is nice. Anyways, I'll be talking today about some of our work in the field of inverse scattering, which is really exciting to us. And hopefully we get some nice discussions going. So the major theme in my group is actually pretty easy to understand, specifically in the field of biomicroscopy. If you have an object that's maybe like a few tens of microns below tissue surface, you can approximate light coming from that object to move in straight lines, and you can use lenses to focus that light and get some really nice images. As that object goes deeper and deeper into tissue, the light starts getting scrambled. And then lenses no longer focus the light for you. And so you have a lot of imaging blur and aberrations and like some sort of like many artifacts related to the scattering of the tissue. So in optical microscopy, this is a big problem because biology occurs at all size scales. So if you have samples that are tens of microns thick, like monolayer cells, or like basically thin samples, microscopy is great for imaging those types of samples at depth. But as you go towards thicker and thicker samples, like clusters, cell clusters, skin, small organisms, once you get to like the millimeter range, then optical methods start struggling and you have to use advanced methods. And then if you get even thicker than that, then typically optical methods are no longer considered the right tool for the job. And so there's this conventional wisdom that if you get too thick, if your sample gets too thick, then the light gets scattered, the information gets lost, and you should no longer use optical methods. So in our lab, the main theme that our lab is centered around is to understand that if you allow light for it to scatter, light can actually travel centimeters through tissue. And so for us, we don't consider this to be an information loss problem. We consider this to be an information scrambling problem. So our viewpoint is like the information is getting scrambled, and can we use computational methods to unscramble that information? And if you can do that, then you can potentially reconstruct the information about the sample that the light is scattering through, and that can be centimeters in depth. So this is an image of just me shining a flashlight through a hand, and you can see the light comes out. And so the idea in our lab is to figure out whether we can use computational methods to unscramble these scattering effects and reconstruct the sample. So this is a hard problem, especially as the sample gets thicker. We've had some like early success in the range of hundreds of microns, which is still pretty challenging for microscopy. And so that is what those are the results that I'll be sharing today. Okay, so the major strategy that we use to accomplish this is to understand that a sample's refractive index completely determines how it scrambles light, how it scatters light. So if you had a way to measure or reconstruct the refractive index of a sample, the 3D refractive index of a sample, then you should be able to computationally undo all the scattering effects, right? And the real beauty of this is that if you can measure the 3D refractive index of a sample, that 3D distribution of refractive index can itself serve as a label-free scatter-corrected image of your sample. And so this field of refractive index imaging is what we spend a lot of our time in. So refractive index tomography, that's what they call it, is actually not new to us. It's existed for a while, for decades at this point, for multiple decades at this point. It's found a lot of early success in fields, in applications where the samples are thin. The reason for that is when the sample is thin, you can approximate the scattering effects. You can approximate the sample to be weakly scattering. And when the sample is weakly scattering, you can derive a linear relationship between the measurements of the sample and the scattering distribution, like the refractive index of the sample, right? And so this has found a lot of success in many fields in biology at this point, and is being like actively used in many applications now. Now, to give you a sense of this, here is a bright field image of a monolayer distribution of cells. Because this distribution of cells is weakly scattering, in the bright field image, you don't see much contrast, right? And that's normal. Now, once you start doing your refractive index reconstruction, all of a sudden you can see some really beautiful 3D morphology of the sample. And remember, this is all based on quantitative label-free refractive index contrast. And you can see the really beautiful morphological features. You can hear we're seeing the internuclear, the texture. We see the cytoplasmic textures as well. We can see a lot of the lipid structures and the vesicles and so on and so forth. So this works really great. Can get really high resolution, sub-micron, maybe around 200 to 300 nanometers of resolution, using simple label-free refractive index contrast. And this is accomplished by simply using an inverse scattering algorithm on this type of weak scattering measurements. So this works great, and this is what's popularly used nowadays. The problem is we care about samples that are typically multicellular. So if this is a sample that is a C. elegans, and the problem is a C. elegans is often considered to be pretty transparent in most biological imaging applications. But even this transparent organism is multiple scattering. And so when you try to reconstruct this organism using the inverse scattering framework that I showed before, you get something that looks like this, right? And so essentially, if you take a close-up, you can see basically no features inside. And so the reason that this happens is because the weak scattering approximation that is required to be fulfilled for the previous algorithm to work is not being maintained here, right? And so then you basically lose visibility of all of your internal features. And the message here is that if you want to image larger and more complex samples, you need to account for multiple scattering, right? And this is the theme of everything that we do now. So this is also not new to us. There's many groups around the world that have their own methods of accounting for multiple scattering. They each have their pros and cons. Typically, I group them into layer by layer, volume integrals, or machine learning. Everything that I'll be talking about today is based on one of our 2019 models, which we call multi-slice. And that's what I'll be talking about today. I'll be dividing my talk into some basic project updates where I talk about the basics of the scattering model. And I'll talk about some of the next steps and challenges as we move forwards. Okay, so let me go over the basics of the scattering model. So there's many scattering models to use. What we do in our scattering model is that we decompose this 3D scattering phenomenon into a layer by layer sequence of 2D scattering events. So typically, if you have the field coming into some layer, right, You propagate that between layers, and then you multiply it by the phase function related to the refractive index of that layer. And that gives you the field that's exiting that layer. And then you repeat per layer until you propagate fully through the sample. So the idea is that if you know the refractive index of the sample in 3 dimensions, and you know the incident field coming in, you can use this computational step to essentially propagate your field in 3D as you travel through the volume, right? And so you can make a prediction of what your measurement will be that exits the sample volume. Okay? In practice, you want to do the opposite, right? In practice, you are measuring the field that comes out of your sample volume, and you want to infer the refractive index of your sample. So we call that solving the inverse problem. And so we do that by using a basic L2 minimization, where we take our measurement and we essentially try to minimize the difference between our measurement and the output of our scattering model for some refractive index distribution. And the goal is to figure out what refractive index distribution minimizes this difference, right? And so that whatever refractive index minimizes that difference between our model and our measurement, that will be our answer. And so because solving for a 3D refractive index from a 2D measurement is ill-posed, we have this L parameter where we try to illuminate the sample at different angles to collect a diversity of measurements. So we try to find a refractive index solution that minimizes that difference across all illumination angles. So that's what this L is for right here. Okay, so in practice, we have an imaging system. We have a scanning mirror that illuminates the sample at different angles. So we basically collect a raw measurement data set that looks like this. So these are C. elegans embryos. These are scattering samples. And we're essentially hitting the sample at different angles. And as we hit the sample at different angles, you can really see this like 3D aspect of the sample rotating. And this is basically showing you that we're encoding 3-dimensional information into 2D measurements. And this is exactly the measurement data set that is fed into our algorithm. Okay. To give you a sense of kind of how this works in computationally, here's a simulation. We simulated a 3D object. We simulated the measurement that we would collect from this 3D object, right? The XY and XZ ground truth values of refractive index are shown here, right? And so when we start our reconstruction, because this is iterative, we have an initial guess. In this case, we have an initial guess of nothing. And then as we run our iterative algorithm, where we're trying to minimize the difference between our measurements and our model output, we see that each of the voxels within this volume starts converging to the true value given by the phantom refractive index, right? And so this is really what we do every time we try to run this algorithm. So it's always an iterative process, and we're trying to match the refractive index. Sorry, we're trying to match the measurements that we get from our system with our model predictions. So we try to implement this in practice for biological measurements, and our hope is that we get similar performance. So here's a kind of a teaser. So this is the C. elegans image that I showed a few slides ago, where we try to reconstruct the refractive index of the C. elegans using weak scattering approaches. And as you can see, we don't get very good results. We use the exact same data and now use this multi-slice approach that I just discussed, and we get an incredibly different reconstruction. Here you can see with really high contrast visibility, all of the really nice high resolution features. You can see the pharynx, you can see the striations along the periphery of the C. elegans, you can see the mouth, all of the high density lipid structures, the muscles, and so on and so forth, right? So this is showing you that, again, the data is the same, but simply by using a different model that now accounts for scattering effects, we're able to reconstruct a lot of these really rich structural features of the sample. So we are aggressively pushing this towards bigger and thicker samples. So this is a intestinal organoid, courtesy of one of our collaborators at UT Austin. Here we can get subcellular, subnuclear features. We can see the nuclear periphery, the nucleoli. This is across around 200 to 300 microns of imaging depth. So this seems to be working well. We are also using zebrafish. So this is a zebrafish, I think around also 200 microns of imaging depth. Again, we get subcellular resolution. We can see really nice structural features, the notochord, the muscle fibers, the floor plate, so on and so forth. So basically, this is just to show that we're trying this on a diversity of samples, and we seem to get some promising results. Up until now, these samples that I've showed have been mostly transparent. They've been scattering, but mostly zero absorption. The really cool thing about refractive index is that refractive index, when you're solving for it, you can actually solve for a complex value refractive index. The algorithm doesn't actually care so much. And so when we image absorptive samples, right, so you can actually see absorption-based features. This is an algae where the chloroplast is absorbing the light that's coming in. If we allow refractive index to be complex valued, remember the imaginary part of refractive index encodes absorptivity. So we can get 3D refractive index, which is the conventional refractive index, as well as 3D absorption-based imaging. And so this is actually really interesting for us now because we're going to try to see if we can use this method to detect absorption-based signatures in scattering structures. So this is kind of where we are right now. This is the stuff that kind of works. But I want to talk a little bit about our next steps and challenges, because I think there's a lot of things to do to push this forward. Our eventual goal is to image something that looks like this, where we have, like in this case, we're typically interested in developmental phenomena. So we really care about seeing processes like this, where you have these really rich dynamic events that are occurring within a sample that's extremely scattering. And so currently, biologists don't have that many ways to study what's happening inside this process in a time-resolved and volumetric way because of all of these scattering effects. And so that's the type of stuff that we want to see, right? And so there's many things many challenges that have to be addressed when trying to see something like this. But the way we look at it is that there's two major challenges. How can we get high-speed volumetric frame rates? Because everything that I've shown you right now is with the sample being static. So how can we achieve high-speed volumetric frame rates of our reconstruction? And how can we image this over large fields of view? Because we really care about getting these system-wide visualizations at whole organism scales. So how can we achieve that? So I'm going to quickly talk about our efforts in these two areas, and hopefully that gives you some ideas of where we're going. So I'll first talk about high-speed volumetric frame rate. Okay, so currently our system is using a scanning mirror that tilts the angle of the beam onto the sample. This is kind of what I showed before. The problem with this method is that to be able to properly reconstruct this 3D sample, we need hundreds of measurements. And the more complex the sample is, the more measurements we need to take to solve that inverse problem, right? And so to give some perspective, for a lot of the reconstruction that I showed earlier, like with the zebrafish or the organoid, we had to collect around 500 measurements. And so the total time to collect all these measurements can be tens of seconds, right? So if your sample is moving at speeds faster than that, then you're in trouble. And so because the acquisition time is dominated by how many measurements we're trying to take, one idea that we have is, can we illuminate the sample with multiple beams at a time? And unfortunately, you cannot do that with this system because a mirror has zero multiplexing capabilities, let's say. So if we want to multiplex multiple angles at a time onto the sample, you cannot use a scanning mirror. A mirror can only illuminate the sample with one angle at a time. And so what can we do about this, right? So one thing that you can look at, one strategy you could expect is, if you look at the system, if you want to illuminate the sample at multiple angles at a time, instead of using a scanning mirror, you could actually think about using a point source array here. So if you get rid of this whole module and you replace that with a point source array, then, and each of these point sources are individually programmable, then that gives you the ability to illuminate the sample at different angles at a time. Each of these point source, depending on the distance that it's offset from this axis, optical axis, will give you a different angle of illumination, right? So this is the strategy that that we're trying to implement. So our current development here is to essentially use a digital micromirror device, which is consisted of an array of individually controllable micromirrors, combined with a microlens array, let's see, to achieve this effect. Specifically, if you image a DMD device onto a microlens array, and you align it so that each individual patch, like individual patches of micromirrors are imaged to individual microlenses, each microlens will now focus to a point, and these focal spots can serve as point sources for on this plane. And essentially, each point source, again, will result in a different angled illumination, right? Because each DMD pixel is individually programmable, you can activate these microlenses separately, independently of each other. So to implement this in practice, we essentially feed in a DMD-based system where we illuminate the DMD with the light source, and illuminate and image the DMD onto a microlens array. And each of the focal spots of the microlens array is on the Fourier plane or the focal plane, and corresponds to an angled plane wave on the sample, right? So now with this system, just based on what we choose, we can either sequentially illuminate each microlens corresponding to sequential illumination, random illumination, so now we don't have to worry about the inertia of the scanning mirror, or what we care about most is multiplex illumination, where we activate different microlenses together, right? And that allows us to illuminate the sample with different angles at a time. Okay? And so this system gives us a unique capability that was not possible with our older system using the scanning-based mirror. To give you a sense of this in action, we imaged a 3D mouse kidney slice, like piece of tissue. And so this is a pretty close-up view. You can see the individual tubules here. And right now, to reconstruct this, we illuminated the sample at 241 angles. So this is showing the Fourier plane. And so basically, this consists of 241 different points, which corresponds to 241 different illumination angles. And if we take a close-up of this, we see a refractive-index reconstruction that looks like this, right? So this is normal. This is kind of the standard technique for us. And the key point here is that now that we have this new hardware system that allows us to multiplex different angles together, now we can start playing around with, can we combine these individual angles into different sets? And that's exactly what we did. What I'm showing here is us multiplexing the angles by a factor 4. So instead of 241 measurements, now we're collecting only 80 measurements. But it still contains all the information, they're just multiplexed together. And what we find is that our algorithm can actually reconstruct a lot of most of the features pretty well. It's not identical. You can see that there is some image degradation. But now the question becomes, can we find an optimum point between how many measurements we have to take and what type of image quality we can achieve, right? And so that's a different question than being forced to just illuminate one angle at a time, because now this gives us more control over how fast we can acquire data. So the obvious next step is to figure out whether we can identify groups of angles that can optimally reconstruct our sample. And that's something that we're actively working on now. We don't have any experimental data on that yet, but hopefully we have some data to share in the coming months about that. Okay, so this is kind of our strategy for achieving high-speed volumetric frame rates. I want to end today with our recent work on achieving large fields of view. So remember, the goal here is to achieve system-wide imaging capabilities, and that typically requires millimeter to centimeter scale fields of view, ideally without losing our resolution too much. And so that's what we'll talk about now. So the major problem with our current system, remember, we essentially illuminate the sample at different angles. We use a lens to focus that light onto our camera. The major problem with this method is that lenses are well known to have a trade-off between field of view and resolution. The larger field of view you want, the more sacrifice you have to have on the resolution part if you're using a lens. So typically, this is why very high resolution imaging lenses have very small fields of view, and very large field of view imaging lenses have very low resolution. Okay? So we don't really want to make that sacrifice. And so our strategy moving forward was to actually remove the lens and then just stick in the camera directly to the sample. And so this way, our optical resolution is still theoretically limited just by free space propagation. So it's not limited by any lens. And our field of view is only limited by the sensor size. Okay? And so now our measurements will typically look like this, where we're collecting, when we illuminate the sample at different angles, because there's no lens to refocus that light, there's going to be a slight defocus gap here. And therefore you see like kind of a sample kind of translating, but it still contains a lot of the 3D information that we talked about earlier. And the image formation process is actually very similar to before. So for now, let's consider two-dimensional samples. So let's say the sample is your X. So the field exiting your sample is X. That field is being modulated by your angular illumination. And then now you have a free space propagation term because it's propagating to the sensor. You take the magnitude squared because you're only measuring intensity. And then you have a pixel binning operation. And this pixel binning operation is important because you're no longer magnifying the image, your resolution for each raw measurement is being limited by the pixel size of your sensor, right? But then that basically concludes the forward model for your for how to predict your measurement, and you try to find a value of your sample field for your phase of the sample that minimizes the difference between your model prediction and your measurements. And if you do your job right, this right here can be converted into an image that looks like this. And the really nice thing about this is that if you get your pixel binning operation correct, your resolution of your reconstruction, this is a phase reconstruction, can actually go beyond the resolution of your sensor pixel. Okay, so we have some pixel super resolution capabilities here. Okay, so our raw measurements typically are collected using a LED array in this case. Just because this is a first generation prototype, this is a popular way to do this. So we essentially get each LED is providing angular illumination onto the sample. And then this is, so I'll show you later our reconstruction. Okay, so the first thing we did was reconstructed a mouse kidney section. So this is centimeter scale piece of tissue. It's optically transparent for now, around 16 microns thick, and we get an image that looks like this. And so we can see that we have centimeter scale fields of view, we have sub-micron, maybe not sub-micron, a micron scale resolution. We have some really nice high contrast features. So this is a optically, like a dense region, structurally dense regions of tissue that we can see we can visualize all of the features with very nice contrast. Again, fully label free. We're pushing this for larger and larger samples. This is a mouse embryo, multiple centimeters of field of view. And we can really see that the diversity of features across the sample that we're able to see because it's a large field of view imaging system. Okay. So everything that I've showed right now is 2 dimensions. We really care about moving to 3D. And the beauty of this approach, so this is kind of the algorithm that we used before. The beauty of this approach is that even though we're considering this to simply be a 2-dimensional field that exits the sample, if we have a 3-dimensional sample, this can simply be replaced by the exit field that is calculated using the exact same approach that I talked about before. So basically, we have our standard multi-slice approach here, and the last field that's exiting the sample can then be put into here, and then everything else remains the same. So we are not really changing our computational model that much. But this allows us to get three-dimensional information. So that's what I'll show you now. We have a different system for this, simply because now that we're in 3 dimensions, we want a larger angular coverage. So we actually, instead of using an LED array, we're actually using a steering arm that illuminates the sample at different angles. And also we're using a laser instead of an LED, so we have more coherence that helps us with the 3D reconstruction as well. And so when we take a measurement, this is a multiple centimeter scale field of view of intralipid, of microspheres in hydrogel, sorry. If you look at a close-up, you can see that it basically looks like very scrambled scattering information. But when we do our inverse scattering algorithm, we get a reconstruction that looks like this. And it's actually really nice, really wonderful. So essentially, these are around 10 micron microspheres that are immersed in around 100 micron thick hydrogel sample. And we are visualizing these microspheres purely based on label-free refractive index contrast without lenses on chip across multiple centimeters with micron scale resolution. And so this is really exciting to us. This is preliminary data for now, but we're trying to see if we can apply this method for thick tissue sections across large fields of view. So we think that this would be a really cool approach for many applications in pathology. So with that, let me conclude my talk. All of the work that I've done today is done by my students, not by me. We're extremely grateful to all of our collaborators who spent a lot of work preparing our samples. I know many groups around the world are really interested in imaging scattering samples and using computational methods to invert scattering, very, very similar to how we do it. So we're very happy to share data. If you're interested in any of the data that I've shown today, please get in touch with me. I'll be happy to share some of our data for you to play around with and everything. So with that, let me conclude the talk. Thank you very much. Thank you for listening.